Synthetic biology is an up-and-coming field. A new technology of taking coded DNA sequences off the computer, creating a DNA code from scratch, and creating a nucleic acid chain is inserted into a blank cell. The DNA sequence takes over the cell, creating an entirely new organism. We also have the technology to place plasmids into other bacteria and have them take in the plasmid and portray the trait. For example, the common pea glow experiment performed by many AP biology students across the nation. The DNA sequence for this radiation-resistant bacteria has been recorded, and so if we combine the methane-producing bacteria with the radiation-resistant bacteria, we can create an organism that could convert the atmosphere on Mars to a hospitable environment, as well as survive and reproduce.
To combine these two very different characteristics is essential to creating a livable environment on Mars. Mars lacks a thick atmosphere, as well as a magnetic field, to protect the planet from solar radiation from the sun. Methane gas has a 72% higher greenhouse effect than carbon dioxide. This is important because Mars' low gravitational force, as well as the incoming solar radiation from the sun, causes a depletion in the atmosphere. Mars' rich CO2 atmosphere must be converted to a mostly methane-rich atmosphere. The methane atmosphere would trap more radiation to heat the planet to a more sustainable temperature. The temperature would sustain liquid water as a byproduct of methane gas production.
The experiment for the Space Lab competition involves attempting to create the combined bacterium in a low-gravity environment and seeing if it can survive as well as reproduce. This organism will be able to survive the cold, lack of oxygen, and the conditions on Mars. It will recreate the environment where liquid water can be sustained, as well as creating a pathway for new life forms. After the planet has been transformed, other organisms can be sent there to jumpstart the evolutionary processes seen on Earth.